Hello and welcome, dear students. How are you all? I welcome you to the class of biology. Dear students, please take your seats. Yeah, okay, thank you. Okay, so this is basically the season of winters, a lot of woolens. So yeah, I'm not feeling well. So you can see I'm tightly packed today. Centers, please report your attendance, and we'll begin right away. Tamenglong and Shillong students, please report your attendance. Good morning, Shillong students. How are you? So yeah, I am not particularly well today. Slight cold and running nose and whatnot. Probably a symptom of influenza. So yeah, so please take good care of yourselves also. Hopefully I should get better by tomorrow. Tamenglong, please report your attendance. Tamenglong students, please report your attendance, and we are beginning right away. So yeah, today I want to talk about in general or a lot of things. So let's not just start with biology today. First of all, today I want to start about some very motivational figures in the history. Motivation for studies, yeah. I want to give about 5 minutes only. I don't wish to spend so much time on it. So yeah, because I'll tell you why later, why I want to discuss this today. Yeah. So first of all, I would like to know from you first. What is the worst thing about preparing for competitive exams? In your eyes, what do you feel that this is the thing that because of which I am lacking in the studies? I just want a small response from you. I'll tell you some things about it later. And I'll start with the studies, but I want to give you a lot of examples how you can improve in a general way. So please tell me first, what is the thing that you feel from your side, yeah, that this is the thing that I'm lacking, this is something which I'm not good at. So it can be a subject, it can be an area of expertise, or an area of lack of expertise. So I want to know what is stopping you. What do you feel that is stopping you? I will surely go back to studies, but first of all, I want to know some things from you. I just felt like I had to take this up today. So better now or never. Yeah. What is that thing that you feel is stopping you? Is some sort of an obstacle that is stopping you from achieving your dreams? Or you feel that this is something like a drawback in you? And one of the most common drawback I feel, it is procrastination. But let us see what you think. That is what I think, me, okay? Now let us see what you think. Not energetic to study. All right, okay, yeah, good. I'll just pen it down. Okay, you are saying not energetic to study. All right. Okay, so anything else you would like to put down now besides this one? Besides, okay, I'll actually discuss all this with you, that what all these things are. Let me put down one more. You have said not energetic to study. So yeah, that is actually a very valid point. In my words, I see this as lack of role model. Role model is a motivational figure. What you are saying, in my word, what I was going to write down actually, motivational figure. Anything else? Okay, yeah. Let's start with this. What do you, okay, so first let's start with not energetic to study. Look, the reason why you don't have energy, reason why you feel like lack of energy, yeah, it is somewhat related to this part. Why? Look, energetic or lack of response, lack of hard work, where does it come from? Where does it come from? It comes from inside us and from seeing the people that are around us. Okay. From seeing the people around us, we generally have this notion that the life is progressing as it is progressing in our immediate neighborhood. Are you getting my point? We have this, like humans have this, we have this tendency to think that whatever is happening around us, everything else will also happen around at that same pace. Now let me give you an example. If you are walking in the darkness, right? Suppose you are walking somewhere in like in a dark forest and a dark pathway. Natural response is fear. Natural response is fear. What if somebody is living in a metro and he is traveling by, let's say, a well illuminated, well lighted pathway, highway in a car with friends? He will not feel afraid. But both people, they are disconnected from the realities of each other. Both these situations are disconnected from the realities of each other. I have given you two extreme examples. If you are feeling lack of energy to study, there will be some people who are very much energetic to study. You must explore the reason why they have this motivation, why they have this in themselves. So I must tell you this, when you are saying that you are feeling a lack to desire to study, and if I am comparing the other end, people who are very energetic to study, so what gives them this motivation? The motivation comes from planning one day ahead. Look, always try to fix small targets. If you are fixing large targets, you will feel lazy, you will feel very much tired, you will feel very much asleep all the time. Okay, yeah, somebody is very tired, I can see in the front desk. Some people, it's okay, no problem. So yeah, that happens, it's okay. So why does this happen? Because we are not planning one day ahead. So if you are always planning one day ahead, always, how do you remedy this situation? Remedy of laziness. Remedy of laziness, or being lazy, just energy lacking. Always live one day at a time. Always live one day at a time. And always plan one evening before. If you're going to do something tomorrow, you should plan it in the night before you go to sleep. Before you go to sleep, at least put this in your mind. Neuroscientists, psychologists, they have conducted a lot of studies and they have found that the best planners, the best people who are, let's say, motivational figures, they have this tendency to plan for short targets. They do not plan for large targets and do a lot of work continuously without stopping. Everybody gets tired. We are human. Everybody gets tired. So why do you feel tired? Because you are not planning well. Because you have not divided your targets into small problems. If you are making some small, easy targets, choose some small amount of time and finish it beforehand. Okay? So if you're going to study tomorrow, let's say, you should at least think about it in your mind. Prepare yourself mentally. Put it in your brain. Put some programmed thoughts in your brain. Your brain will be ready for the next day. If you're planning just before it, it becomes haphazard, right? So that is what happens. Okay, so it's very easy to get rid of this problem. But all that it requires is you should plan one night ahead that I am going to study this tomorrow and let us see how it goes. You should plan something and you will definitely see the results next day. Then you should plan with better preparation, with better motivation. So this motivation will come only when you fix some small targets and you complete them. This will give you the confidence to move further. You will fix a better target and something which can be completed, which is feasible. I do not want you to take 100 to 200 pages and finish it once and for all. I want you to finish at least 50 questions, 20 pages. That is something which is very achievable. Per subject, you can take 20 pages. If you are studying one subject for a very long hours, 6 hours, you cannot study at all. You cannot do that because your brain has different regions. Our brain has different regions. One part is for thinking logically and that part also has many centers. For solving physics, you need to do work from a different part of your brain. For learning things in biology, for example, you use different neurons and some neurons are common. Some are common. But you do use a different region of your brain. In chemistry, it's somewhere between the two. You have to employ both neurons, learning and innovating. So one part is creative thinking. Physics requires creative thinking. Biology requires small bit of creative thinking, and most part is memorizing these concepts for using later on. That is what has to be done. So if you have to learn something, imagine it first. So if you see any motivational speaker in general, they will first ask you to dream. What is dreaming? It is setting a small target. So always achieve a small target. And then now let's also understand this with the help of a better example. Everybody needs a role model. You need a role model. You need someone to energize you, to help you out, to meet you personally, to give you time and hear your problems out. So if you go to the church, you can find some people there. If you are a studious person, you must have some person in your life who can at least act as a role model. Your role model can be your teacher. It can be me. I won't place myself in that category, sorry. You can, but yeah, it's a difficult thing to do. You can, of course. It can be anyone. I prefer, if I had to choose a role model, I would choose someone I know personally and who lives by example. He says something and he also achieves that. He also does that, what he's saying. That should be exactly the same. There should be no contradiction among it. That is some person you can call a role model. So role model should not necessarily be associated with your field of subject. He should not be necessarily a doctor or engineer. He can be, but he also cannot be. It can be anyone who has the capability to achieve targets, who is someone in an administrative capacity. It can be a principal, it can be a brother, it can be a sister, it can be a cousin also. It can be anyone. But you have to choose this person carefully, because really responsible people, really good people, they are very rare to find. Most people are just good at saying things, but not necessarily achieving them. So you have to choose one person carefully. You can also choose one person for a short time, and then you can change your role model, because you feel somebody is better able and better suited to guide you through it. So you can learn by experience, okay, and hit and trial method also works. So you need to choose someone in your life, someone you can talk to, right? Someone you can talk to, seek advice, listen, okay. And basically, if you want to become a great person in your life, you will have to listen to a lot of people. You should learn to listen to a lot of people, and that is something that you will carry throughout your life. It can be something of a game changer to you. Okay. So in general, what we are lacking in ourselves, that can be made through by seeking help from the others. Never be shy in seeking help. Always try to get some advice. Always try to follow it up to some extent at least. If you complete some part of an objective, it will give you great satisfaction. If you complete no part of it, it somehow goes into our mental status, it goes somewhere in our mind, and it basically stings us that we did not do this, let us do something else to get away from this responsibility. Okay, so that's not what you are looking for. That is not something which you should actually hope for. Okay, pleasure is different from happiness. You complete targets and that gives you happiness. You complete responsibilities, you take responsibilities, it gives you respect and happiness. And respect is gained, first you have to respect yourself, and self-respect is only gained, it cannot be given. Nobody can give you self-respect. Self-respect you can only gain through one way, by completing your responsibilities. And that will, believe me, it will give you a great amount of satisfaction. It will give you a lot of happiness from the inside. Okay. So, find some people, talk to them, and always try to cooperate with each other. Show some kindness to people, to animals, and this will go a long way in your life. Okay. So I want to give you very little advice today, because I think that too much of advice, that will basically bore you out. So in let's say in about next week or tomorrow, whatever day you prefer, I'll talk about procrastination. Okay, I do not want to waste too much time on advice. A short piece of advice, something which you can follow, that is only necessary. Okay, so the next class, whenever we have it, we'll talk about procrastination, and that will be something else. Okay, so right now I told you some things. I hope that helps you out. Okay, so start somewhere. Do not wait for something. Do not wait for the right time. Start now. Start right now. Okay, so go home. When you go home today, okay, after the classes, try to find some motivation. There has to be some motivation. It is somewhere around us. Okay, you will find it. Just have to listen to your inner voice. Okay. All right. So much for lectures on philosophical topics. Let us get back to our studies. Okay, and okay. So I just want a small bit of feedback from you. In biology, in particularly botany, how much questions have you completed on your own? Look, I have completed the living world. I have completed biological classification. Okay. I have completed plant kingdom. Okay. Now, do you first let us discuss these three. Do you face any difficulties in any of these three chapters? After this, I will discuss the chapters that come for after this. So, do you face any difficulty in any of these chapters? Do you want more resources from my side or notes from my side? Anything else? What questions were you facing the difficulties on, if I may ask? For example, if you have completed living world, there is not much in living world. In biological classification, there are 5 kingdoms. So, is there any kingdom that confuses you, any part that confuses you? Are the classification categories or the classification criteria, the systems of classification, natural, artificial, is there anything in this chapter that confuses you, or is it plant kingdom that confuses you? Okay. You are saying that biological classification chapter you need more assignment. Okay, done. Okay, so let me do this tomorrow or today. Okay, I'll do this. By the end of this week, you will get assignments for biological classification. I can give you plant kingdom also after that, but first I'll give you assignments and questions. Basically, there will be a lot of questions, MCQs for biological classification. Okay, done. That also you will get. Okay, let me send both of these to you. I'll give this to you with a 1-day gap, so that at least you can do one thing first and then move on to the next. Because if I give you all of them together, you might feel a lot of pressure on yourself. Okay, so I don't want to pressurize you. First, I will give you biological classification. I have the questions, so I'll do this first. Okay, plant kingdom, I'll do that also. Anything else? I have given you a lot of PPTs also for both biological classification and plant kingdom. Do you want me to give more PPTs, any of these chapters? Because you observe that the questions are also out of book. Okay, yeah, the questions are also out of the book. Yeah, okay, sure. Yeah, there are a lot of questions like that. I don't contest that. Okay. Alright, so okay, I'll just give you the assignments and I'll give you the answers along with it. Assignments I'll give you for both the chapters. Okay. So by the end of this week, you will receive both. Okay. After this unit, do you face any question? Any doubts on this regard? After plant kingdom, what difficulties are you facing? Look, plant kingdom, basically you have to discuss about algae, bryophyta, pteridophyta, gymnosperm, angiosperm. So that should not be much of a problem, but let me know if you have anything else. Okay, morphology. So morphology I am already continuing. So this is running part. By the end of that morphology, you will receive a lot of questions. There are a lot of questions from morphology. Morphology is, in my opinion, a very interesting part of biology. So I'll take care of this, that you do not face any difficulty in this. Okay. Alright, so let's continue. Let us start with where we left off in the last class. Okay. I believe we were, yeah, just a second. There were a lot of things I had done in the last class. Okay. Can you send more examples for morphology? Okay, okay, sure. Morphology, you want more examples? Okay, so basically I'll have to give you PPTs. I'll have to give you PPTs for biology, morphology, yeah. So what I will do is I'll make some PPTs for you with a lot of pictures, and for example, if storage root is there, so I'll give you the example of carrots and all that, like carrot, all those examples. There are a lot of other examples also. So I'll make PPTs for you for the morphology part. Okay. Anything else? Anything else? Yeah. Any other chapter from for the droppers part, like any other difficulty that you face? Look, I have to cover a lot of things. I hope I am going at a right pace. You can name other chapters also if you are facing any difficulty from that. You can name other chapters also. Okay. Look, we are all almost on the verge of, let us say, morphology, anatomy. Let me write the name of the chapters: anatomy of flowering plants, morphology of flowering plants. Morphology, I'll give you PPT. Okay, yeah, that's good. Small target. Let us do this first, and then we'll move on to the next parts. Continuing from where we left off in the last class, we will have to start. Like we had finished the Krebs cycle, and we had completed the plant growth hormones almost. Okay. So plant growth regulators we were doing. Okay. So we were basically discussing the plant hormones. Okay, just a second. Okay. Let us continue from that only. All right. So plant growth regulators, this was what we were doing. Yeah, I was actually looking for that exact part where we had left off, cytokinin. Yeah. We had finished auxin. Auxin we have already finished. So auxin, I told you a lot of things. It controls phototropism. I gave you the example of sunflower. I gave you the example of lotus. I gave you the example of tendril coiling around the support. Okay. Yeah. Thanks for reminding me. Yeah. Auxin we have already completed. Auxin's most important response is phototropic response. Also, auxin is responsible for initiation of root, suppressing the growth of weeds in a agricultural field, and yeah, there are a lot of other things also, but let us not go into those details right now. So basically auxin is phototropic hormone that is controlling a lot of those things. Okay. So let us start with where we left off, that is basically cytokine. Cytokinin, I had told you, it is synthesized in the root. Cytokinin synthesized by the root. Root cells synthesize cytokinin. Cytokinin, you can not say that it is exactly opposite to auxin, but there is one behavior of cytokinin that behaves in different fashion to auxin. Auxin causes growth in the vertical direction only. It causes plants to become tall if auxin is only applied. Cytokinin promotes the opposite response. Cytokinin causes growth of lateral shoots. Okay. Cytokinin has a long history. We had discussed that cytokinin, it was isolated from herring sperm DNA. It was isolated from a lot of other examples also. It was isolated from corn milk, coconut milk. Okay, there are a few other examples also. So basically, the attempt of this exercise was to isolate a lot of plant growth regulators from different factors, and they all had the same effect. They increase the cell division rate. You put a cytokinin hormone in a plant tissue, it immediately starts to perform cell division. So that is why this name was given, cytokinin. Cell division rate increases the rate of cell division. It is going to occur very fast. So kinin means fast, cyto means cell. It has a profound effect. It was isolated from different sources, such as herring sperm DNA. Actually, herring sperm DNA, it was denatured, not fresh sperm DNA. The herring sperm DNA was basically kept for some time, and after it started decomposing, it decomposed into a derivative of purine nucleotides. It is called 6-phosphorylaminopurine. Aminopurines are the general category or the chemical name of cytokinins only. So by chance that resembled the cytokinin of plant cells. By chance. Herring sperm DNA, normally it is found in some plant extracts and some plant extracts such as maize seed extract. You can see this maize seed extract. Take a corn which is not fully mature. Let's say we take a stage somewhere between baby corn and a full grown corn. Okay, somewhere between that. So the corn is not fully ripe, so the seeds are not very hard. If you pluck a small seed from there and press it between your fingers, a milky substance will come out. Okay, this is the sweet corn, and that extract contains kinetin or zeatin. Zeatin, zea maize. Zeatin and coconut milk kinetin. Later on, after analyzing the structure, it was found all three substances are same. They are derivatives of nucleotides called aminopurines. Later on, all three substances were found to be similar, were found to be same, called cytokinin, and the chemical name of cytokinin is 6-for-furyl aminopurin. Six-for-furyl aminopurin. Okay. This is about cytokinin. Now let us study the effects of cytokinin. Effects of cytokinin. Cytokinin is comparatively very easy. Auxin is very long. A lot of questions are asked from auxin. Their scope is too high. But when we talk about cytokinin, very few questions are formed, and it has one or two main effects that we are going to study. Effect of cytokinin. So when you look at your school garden, there must be a school garden that you have. Very sorry. I have this sneezing problem today. Sorry. So what happens is, gardeners, let us talk about a small bush. If bushes are allowed to grow by themselves, they will grow in this fashion. They will grow vertically tall. Very less leaves will be found on the lateral branches. The lateral branches are mainly undeveloped. It is growing straight. Most of the trees, without proper care, they will go straight tall, and that is a bit of problem. Trees which are tall face a lot of resistance from the wind. If they grow tall very fast, there is a problem. If some thing happens like wind velocity is too high, they can drop down easily. Branches will break. If branches break, that means the fruits, leaves, a lot of things will be lost. Okay, and the root system, if it uproots, it will take a lot of plants with itself. So basically, a lot of damage will be done. So whenever the people are allowing the trees to grow in their garden, they will take proper care of them. They are giving fertilizers to them, so that means trees will grow fast. In the jungle, they grow very slow, so that is why their trees are very strong. But when we give fertilizer and a lot of things, the trees grow fast. Now their growth has to be regulated. Cytokinin has the effect of suppressing the apical growth. The farmers, the agricultural experts, they ask to cut off the apical portion. Cut off the apical leaves, cut off the apical bud, not the apical leaves, sorry, apical buds, and that includes a lot of leaves along with it, and then the lateral growth will happen. Plant will spread in all directions. It will spread in all directions. So effect of cytokinin is opposed by auxin. Cytokinin promotes growth of lateral buds. Lateral buds. It promotes the growth of lateral buds. In presence of auxin, it is suppressed. Auxin basically suppresses cytokinin effect. The effect is suppressed. This suppression of lateral buds due to auxin is called apical dominance. Auxin causes apical dominance. Now what the gardeners do, they cut off the top, they take a big knife, they take a, sorry, not the knife only, they take a big scissor and keep on cutting the topmost branches containing the apical buds. So when they cut that, plants have a bushy appearance. They grow in all directions. Having a bushy appearance means more green appearance. So that is what they do. Gardeners therefore keep on pruning the branches, keep on pruning the branches of plants in orchards and gardens to make plants grow uniformly in all directions. Uniformly in all directions. Okay. Okay. So that is what is done. So basically they keep on cutting the top branches, so the side branches they will continue to grow uniformly. Because auxin is suppressed, now cytokinin will show the effect of itself, but in presence of auxin it cannot show its effect because auxin dominates over cytokinin. Okay. So plants get a bushy appearance of plants in garden. Okay. Now, besides all this, besides observing this fact, there is one more very practical example of cytokinin, and that is a very important one. Tissue culture. Tissue culture. How many people are aware of tissue culture? How many people of you are aware what tissue culture means, or they have actually observed a plant growing in a test tube, a very small plant? Yeah, some people have seen. I'm very happy. Yeah, very good. Yeah, you can visit your local nursery or a scientific institution and they will give you small plants. You can ask them, and most of the scientists working in botanical organizations, if you ask them, they can give it to students very carefully. Yeah, they will give you a few instructions how to take care of the plants, and normally you cannot grow them directly into the soil. The tissue culture plants which are growing in the test tube, they need to be put in a greenhouse so that plants can be adjusted. Okay, so that plant is comparable to the thing is, if a plant is growing in a test tube, it cannot directly be transferred into a sunlight and microbes and soil. It will be spoiled immediately. It cannot adjust because in the test tube it is getting everything, and suddenly it is being made work so hard. So first of all, we transfer it in a greenhouse, let it grow for some time, and then we can do that. But if you visit a scientific facility, they will show you lots of plants growing in test tubes. I'll give you a very practical example. In nurseries, nowadays they have a variety of banana that is grown by tissue culture. if you plant a normal kind of banana plant, it will give fruits in 6 months. 6 months. But if you use the tissue culture variety or the tissue culture plant, it will give fruits only in 3 months. So the farmers are now preferring the tissue culture produced banana because that same plant is giving fruit in 3 months. So in 6 months they will have 2 crops of bananas. Okay, so that is very helpful. So a lot of things happen. Also in the northeast, in your region, there are very, very rare orchids. Very, very rare orchids. And orchids have a problem. They have very, very minute seeds with little or zero endosperm. They are endospermic seeds, but the quantity of endosperm is so less so that if these seeds do not germinate very quickly, they will die. So very rare orchids, their seeds are put in a test tube and grown by tissue culture. If tissue culture is not used, many, many rare orchids, super rare orchids, they will become extinct. Now let us understand what tissue culture is. If I am to apply tissue culture to your body, I can call, let's say, the girl sitting in the front. Okay, I can take your hair. I will ask you to come towards me, and I can take one of your hair, and I can put this hair, supposedly in the future, in a growth medium, and I can grow exact clone of you. That is what is exactly meant by tissue culture. To grow a genetically exactly similar organism from which a cell or part of tissue is derived. Your hair follicle, if I pluck your hair from your head, hair contains a bulb-like structure at the end. It is called hair follicle. The rest of the hair is consisting of dead cells, but at one end there are live cells. That is used in DNA fingerprinting also. Okay, and these things in the future can be the basis of cloning. They can take some part of your body, maybe. Right now it's science fiction. Right now it cannot be done. For example, I saw somebody, he was a pseudo-scientist. He was saying that it was a very funny thing to watch, a very funny interview. He was saying that the President of America does not touch a glass of water, and if he touches a glass of water like that in any other country, the FBI and the CIA, they immediately clean the glass with the help of some tissue paper or something, so that his cells in his skin cannot be used in the form of tissue culture by any other man, and he can ask that clone to give all the information in his brain, okay, regarding all the secrets of America. So that is super science fiction, and that is not even science fiction. It is the complete bogus thing. So I saw such funny interviews some time ago. That cannot happen. But the science fiction can, in general, we have some principles of cloning, like Dolly, Molly, Polly, they were created by cloning. So we have some part of tissue taken from them, and they can replicate the exact organism. However, if you make a clone of somebody, that does not mean that their memories will also be the same. Memory is a different thing. Memory cannot be created by cloning. It is experienced differently by different organisms. Now, tissue culture, the principle of tissue culture in this world was given by Haberland. Haberland first proposed the principle of tissue culture. This is important. This name is important. Normally in NEET questions, names are not asked. Names of scientists are totally not asked. And there are very, very few exceptions. This is an exception. Haberland was a German scientist. He gave the principle of tissue culture, proposed the principle of tissue culture, and based on this principle we have the principle of tissue culture in animals. tissue culture in plants. Culture in plants. He proposed the theory of totipotency. Theory of totipotency in plants. Totipotency is a very, very important term. It is a very important term. Theory of totipotency. Totipotency in plants. He talked only about plants, nothing else. Just a second. Okay. Now, what is totipotency? Totipotency, that means that any plant cell taken from a plant has the potential to grow, to undergo cell division and grow into a full-fledged plant. That is the principle of totipotency. So, Haberland proposed that if I take a plant cell from, let's say, the leaf cell, and I put it in a suitable growth medium, he proposed that this should be able to grow into full plant, and he proposed this for carrot, for example. He did not have the technology to do it, he did not have the resources to do it, he did not know the exact nutrients that are required with the plant and how he should give it, he did not know. He was very much ahead of his time. He proposed this principle in 1920 at the time of near the time of First World War, and at that time where there were no scientific resources of what he required. So, about 50 years later, 60 years later, his dream was realized when there were other scientists who put this theory into action. Totipotency. Totipotency, it is the ability of any plant cell to be able to grow into a full plant by cell division and differentiation. So, any plant cell can undergo many multiple cell divisions and differentiation to grow into a full plant. This is the principle of totipotency. So, I am going to draw the diagram of tissue culture now. He proposed this for totipotency, that this should be the principle of totipotency, and let us explain, let us try to find out what tissue culture is all about. You can just say that tissue culture is based upon totipotency principle. Now, what he proposed was, let us take a plant cell, single cell. Let us take a single plant cell, leaf cell. This is carrot, leaf cell. This is the nucleus. There are many chloroplasts here and there. Carrot leaf cell. Carrot leaf cell is taken and put in a test tube containing cellular growth medium. Cell growth medium. Basically, it has glucose, sucrose, basically sugars, proteins, carbohydrates are not required. Basically, sugars are required and inorganic nutrients are required. Plants can manufacture everything by themselves. So, cell growth medium or nutrients in general. Just you can say nutrients for now. Already you have studied mineral nutrition, you know what mineral nutrients are required by plants. So, we will prepare a solution of it in agar-agar. In agar-agar. Agar-agar is derived from, do you remember? Yeah, please scroll down again. Yeah, sure. Yeah, let me know when you are finished, okay? Let me know when you are finished. I'll scroll more. Okay? When you are finished, please let me know. I'll move on to the diagram. I didn't know that I was writing so fast. It's okay. I think you must have completed the previous one. Please write fast. We have done it. Okay. So, cell growth medium in agar-agar. Do you remember agar-agar is derived from which group of plants? Red algae. Do you remember the name of the red algae from which agar-agar is derived? Gelidium and Gracilaria. Just trying to help you remember. Now, this plant cell, when it is kept in agar-agar, it will undergo de-differentiation. De-differentiation. Do you understand what de-differentiation is? In anatomy of flowering plants, we discussed about de-differentiation. How many people of you remember it? Say yes or no, or raise your hands if you do not understand de-differentiation. If you do not understand, raise your hands. Okay, you do not understand. De-differentiation means, yeah, okay, I understand that you are forgetting it. It's okay, I'll repeat it. Look, de-differentiation is the opposite of differentiation. Differentiation means something is losing its original identity and becoming specialized to perform a special function. For example, you were in class 10th, then you took science. You were also studying maths along with it. You were a science student. But after 10th, you became specialized to become a science student. You took biology. Or somebody took maths. That is also science student. But you are now going to specialize towards the medical field. You will be called a doctor in some years. Okay. The one who takes engineering, he is specialized for engineering subjects. So this is specialization. If I take you and I turn you into a baby, if I take you and turn you into a baby, now you are de-differentiated. You will choose your subjects from class nursery. that yeah, I have to take this subject, I have to take this subject, and from 10th class you can make your decision again that you want to take maths or science. So this is dedifferentiation. A cell loses its identity and it becomes a meristematic cell like an embryo cell or a zygote cell. This cell is going to undergo very rapid cell division. It is not a leaf cell anymore. It is becoming a meristematic cell. Now you understand? Just don't raise your hands if you understand it. If you understand it, yeah, okay. If you understand it, raise your hands. That's what I meant. If you don't understand, don't raise your hands. That means that if you understand, you must raise your hands. Okay. Okay, still some doubts among people. Yeah. Okay, boys understand. And some people are writing. It's okay. Look, I'll repeat again. By dedifferentiation, I mean a cell is going to become like a baby, like a rapidly dividing cell. And when it converts again into a root cell, into a leaf cell, into a stem cell, or into a flower cell, this is differentiation. Dedifferentiation means becoming a rapidly dividing cell, becoming meristematic again. Cell growth medium in agar-agar. Now this is a different undifferentiated cell, and in agar-agar I have one particular thing, cytokinin upon auxin ratio. Auxin upon cytokinin ratio is equal to 1, or you can say auxin is equal to cytokinin. In this cell growth medium, which contains nutrients, I am putting some auxin, I am putting some cytokinin. I am keeping the ratio equal to 1. So when auxin and cytokinin are same, rapid cell division will happen. Rapid cell division and after the results are analyzed of the cell division, I will see that the cells have formed irregular cellular mass growing in all directions. Irregular cell mass. Irregular cell mass. It is also called callus. Callus. Callus is an irregular cell mass of undifferentiated cells. Callus represents undifferentiated cells. Undifferentiated cells. Undifferentiated cells. Callus. Now I am going to take this one step further. Now I am going to increase the auxin and decrease the cytokinin. Or you can say either increase auxin or decrease cytokinin so that the ratio becomes greater than 1. Auxin should be more than cytokinin. In this condition, callus will grow root. I have omitted one step here. Callus is then cut into small pieces. Each small piece is put into separate test tubes. Cut into pieces like this, like this, like this. So put it into different test tubes. Now take one test tube take one test tube and increase oxygen so that oxygen upon cytokinin ratio becomes more than 1. Under this condition, callus will grow, root will grow like this. Root will grow. When oxygen is more, I told you that oxygen causes growth of root or development of root. Not exactly growth. Oxygen is responsible for root initiation. So increase oxygen, you will get root. If root is not developed, plant will develop root. Now you will perform another experiment. Now you will decrease the oxygen and increase the cytokinin so that oxygen upon cytokinin ratio becomes less than 1. When you do this, now what will happen is under this condition, shoot growth will happen. Now shoot growth will happen. Shoot growth will happen. This is a small miniature plant. I have drawn it a bit larger. Actually, it will be very small. If you look in the test tube, you will get a very, very small mini plant. Very small mini plant is there. I am telling you this because I have seen this tissue culture happening in front of my eyes. Actually, for your experience, I have drawn this test tube very small. Otherwise, all this experiment is happening inside the test tube or the petri dish, and in the initial stage, very small plant develops. As you continue the experiment, after some time, the plant will start growing out of the test tube. Okay, this is happening. So when you change oxygen cytokinin ratio, now shoot development will happen. Now you transfer to greenhouse for hardening. What is hardening? Suppose you take a millionaire boy and you put him in the slums of Mumbai. Take a millionaire boy, somebody, let's take some person like Barack Obama's daughters. Let's take some boys like you can take any example. I can let's say take Justin Bieber. Okay, let's take Justin Bieber and put him in the slums of Mumbai. So he will immediately collapse. He cannot survive those conditions. So that is a similar case with plants. A plant which is growing in very protected environment, lots of facilities, it cannot survive immediately if you put it in the soil. So what you can do is, you can wait for some time, you can wait for it to adapt to the soil conditions by putting it in a protected greenhouse environment. Something which happens in growth, it's like a mediator, middle ground, middle environment between soil and between test tube. So this is called hardening. When it has learned to grow in the greenhouse, now it can survive in the soil. Now you can transfer it to soil. Transfer to soil. This is called tissue culture. I took a plant cell. I'm making many plants. Actually, callus is cut into 20, 25, 30, 50 pieces. Each of these 50 pieces, let's take 8 to 20 pieces. From one cell, you are getting 20 plants. This is tissue culture. This is the magic of tissue culture. Taking a small amount of plant tissue, you can grow as many plants as you want. This is tissue culture. Please tell me, let me know just as you have finished tissue culture. Okay, have you done it? Let me know as soon as you are finished. I think everybody has drawn it. Okay, so let us continue to gibberellins. Everybody, I can see it is, yeah, everybody is a bit tired and sleepy. So winter is one such time when you don't want to put your hands on the table, you don't want to put your hands out of your jackets. I can see how many people are just taking your hand and putting in your jacket. So I can feel your pain. I have been through that and I also face that problem. In fact, I have to teach and so many times, yeah, my hands are always out. So yeah, I understand your pain. Sitting on the benches and trying to write down, take some notes. It's a real pain in winters, right? I can feel you there. Okay, just to give me a moment, I need to give you some solid examples in this regard. Yeah, so we were actually, yeah, give me a second. Okay, so we had done this, this is also done, this is also done, but I wanted to give you some examples of some beautiful grapes that I wanted to show you. And they were actually being grown by okay, this is algin. This is algin experiment. Alginate obtained. I think I've shown this to you. Brown algae. So some people are putting their hands in a sticky substance, that is algin. Thought it was something else. Yeah, okay, they are not here. All right. Actually, I believe it's because of a virus infection, I guess. Yeah, okay, I found it. Yeah, one of my folders got deleted. Okay. So let me show you some beautiful images that will help you in understanding what gibberellin is all about. Okay, found that. And I need to give you more examples. Give me one more minute. I am almost done with this here. 59.832. 59. Okay, yeah. So let us take a look at these pictures that are here. Do you see some beautiful grapes here? Do you see some beautiful grapes here? All these grapes are very, very large in size. It's a particular variety of grapes. One is called Indian. One is an Indian variety called Sultana. Other is a variety called Thompson seedless grapes. and you can see these are very large actually, and these are very juicy also. Yeah, this particular variety of grapes is very juicy, and it has a lot of advantages to itself. So what happens is, yeah, just give me a second. Just please wait. Okay. Okay, I'll continue a bit. So what happens is, these seedless grapes that you're seeing here, they were cultivated by the application of a plant hormone called gibberellin. Gibberellin has the property of increasing the metabolism of the soluble and the insoluble substances, and everything is converted into a monomer or a dimer, and most cases it is a dimer, and it is called, the dimer is called sucrose. Okay, it's a disaccharide actually, not an actual dimer. Gibberellin increases synthesis of sucrose from starch. So basically, when we are talking about a plant, it is going to convert all the sucrose, all the other substances into the sucrose. Okay, and I want to show you some other examples here also. Just please wait. Please wait, there is a issue, I guess. Please wait for 1 minute. Yeah, okay. So I want to show you I guess there is a okay. In the meantime, let me show you some other examples. Yeah. Okay. Yeah, there is an issue. Please wait. Okay. Now let us see some more examples here. These grapes that you are seeing here, yeah, okay, 873. this is one more picture. These grapes are actually being measured by an instrument that gives an idea how much large the grapes are. So this is the largest grape size that is available on the market. You can see that there is a small some sort of a scale there. So from the smallest to the largest grapes available on the market in India or elsewhere. Actually this is not of India, but these grapes are imported in India, and I bought these grapes. It's a variety of grapes called Sultana. So it's grown in the Middle East and basically Afghanistan. That's where it actually comes from, but it has been developed by Indian scientists. So yeah, nowadays being cultivated in India also in some parts. So this is the largest grape size available on the market, and there are some other examples also. Now gibberellin, whenever it is sprayed on any plant, let it be cabbage, let it be maize, let it be wheat, rice, anything. Whenever it is sprayed on a plant, it causes the plant to become tall, it causes the size of fruit, size of seeds to become larger, and this causes the fruit to get a better price on the market, because larger, juicier, sweeter fruits are always going to get a better market share. So normally the fruit that is most used is grape. We have experimented on apple, we have experimented on pear. Lot of fruits are being experimented for the effect of gibberellin, but the fruit which has developed most sugar after spraying gibberellin is grapes. So that is why it is being used for that. Gibberellin has the general effect of causing parthenocarpy in fruits, in plants, Auxin and gibberellin are parthenocarpy inducing hormones. So that it means if you spray these hormones on the plant, it will develop the fruit without developing seeds. Parthenocarpy is the development of fruit without the actual development of seeds. No fertilization, just the ovary wall becomes mature, and mature ovary wall is called fruit. So fruit development without seeds. That is the property of gibberellin. It has the property to induce bolting. How many people are aware of bolting? Bolting you have to see in cabbage plants. In cabbage what happens is, in the winter season, the cabbage is tightly packed. All the leaves are overlapping with each other. The internode are very compact. But if you spray gibberellin, it will suddenly grow tall and all the leaves will open up like this. This phenomenon is called bolting. And on the top of that plant, cabbage plant, flowers will appear. So if you leave cabbage, if you do not eat the cabbage, if you do not take it out of the soil, in that case, in the month of February, natural gibberellin development in cabbage takes place. So bolting is done in February, but if you spray the gibberellin right now, in the month of December, it will begin to show bolting. Any plant which is a short day plant or a long day plant, okay, it will change its behavior when gibberellin is sprayed. Gibberellin causes flowering and fruiting. So this is a flowering fruiting hormone. Can you tell me the name of the hormone which is exactly opposite to gibberellin? Abscisic acid. Abscisic acid is the opposite of gibberellin. Abscisic acid causes the plant to undergo dormancy. It wants to go to sleep. That I am in the sleep mode. I will not work. It's the adverse condition. Winters are coming. Let me get a good blanket. Let me drop all my leaves, and let's go into dormancy stage. So that is the effect of abscisic acid. Gibberellin is the opposite of it. It causes flowering, fruiting, sugar development, okay, all these things. So gibberellin is the opposite of what you have studied up till now. Let me show you few things about gibberellin. I guess you must have already studied about gibberellin in the PowerPoint presentation that I have given you. This is the chemical structure of gibberellin. It is 3-gibberellic acid or Ga3. Where it was discovered? It was discovered in Foolish-Bickeney disease. In the Foolish-Bickeney disease, what happens is, in Japan in the 1930s, a strange disease was observed in rice plants. They become abnormally tall. And what will happen if they become tall? They will fall off. If they fall off, no fruit, no seeds. So this is a foolish seedling disease. In Japanese, you call it Bickeney disease or Bickeney disease. It was later on discovered that there is this disease due to fungus called Gibberella fujikuri. Gibberella fujikuri is producing gibberellic acid. It is similar to plant hormone. So this plant hormone is causing the seeds to become tall. Okay. So it causes this disease, and it is synthesized in apical portion of stem and root. That is apical meristem, root apical meristem, shoot apical meristem. In class 12, you have genetics. Do you remember Mendel's dwarf plants, Mendel's tall plants? Say yes, raise your hands. If you remember Mendel's dwarf plants, Mendel's dwarf and tall pea plants. Nobody remembers. Okay. I guess you don't want to take your hands out of your jackets. Alright. Mendel's dwarf plants can be converted into, it's an important question in class 12. It is related to gibberellin. Mendel selected pea plant and he had some specific characters for it. What happens is, if you take a tall plant, if you take a dwarf plant, and if you spray gibberellin on the dwarf plant, it will become tall like the capital T plant, same height. So why does this capital T and small t, what is the effect of these genes, alleles? The effect of small t is that it produces less gibberellin. This I will teach you that when the class 12th topic comes. So you remember that dwarf and tall plants. Dwarf plant can be converted by spraying gibberellin hormone. It will become tall. It's a strange thing and it's an important question. What is the difference between capital T and small t genes in terms of plant hormones? Small t produces less gibberellin, capital T produces large amount of gibberellin. So that is why it is very much tall. That is the reason. Continuing, gibberellin causes cell elongation, okay, and also causes cell division. It breaks the dormancy in seeds and birds and grasses wherever. It causes flowering, okay. And these are the, on the left hand side, these are the grapes produced without gibberellin. On the right hand side, these are the grapes produced by spraying gibberellin. Can you see the difference between the grapes? It is visible to everybody. Difference with and without gibberellin. You can see the, also one thing cannot be seen in this figure. It is the taste. The right hand side grapes are sweeter. They are very much sweeter. Left hand side grapes are sour. You remember the fox and the grape story? Grapes are sour. So that is why I cannot reach them, right? This is the Thompson seedless grapes grown in California. They are imported in India also. Now bolting. If you spray gibberellin on a plant, it will suddenly grow flowers on it. Okay, so this is called bolting. So previously it was not having flowers. Now suddenly it is growing tall and producing flowers and all that. Without gibberellin, with gibberellin, it is growing tall, producing flowers on the top. On the top of that is the flowering axis, inflorescence. Okay, so gibberellin has the ability to do all this. Also, in making beer, what we do is either we can spray some gibberellins on the germinating barley seeds. If you do that, all the starch will be quickly converted into maltose, and fermented malt is called beer. Okay, so you want to do that quickly because then the yeast will produce the alcohol faster. That is the thing. So mobilization of food reserve, that means conversion of starch into sugar. So now we can come back. Come back on this part. I'm inserting one more picture here that should be interesting to you. Yeah, see this, this is not peach or plum, this is grape. You can see the size of grapes bursting with lot of food, lot of juice inside them. Okay. So you can write, gibberellin was discovered from Gibberella fugiculae, foolish seedling disease. Foolish seedling disease causes Bacony disease. This is the diagram of that. This part. This is normal rice seedling. You can write. Gibberellin was discovered by two scientists, Yabuta and Sumuki. Gibberellin was discovered by Yabuta and Sumuki while working on foolish seedling disease. While working on Bacony disease. Foolish seedling disease. Foolish seedling disease. So they discovered that it was due to infection of a fungus called Gibberella fugiculae. Producing GA3, gibberellic acid 3, gibberellin hormone. So what they did was, they said that this is happening due to production of substance by a fungus. So this fungus that is producing this substance, let us see what effect it has on other plants besides rice. So they found that it is fascinating. If you spray it on grapes, grapes become larger. Spray it on any plant, it becomes larger. Spray it on any plant which is dormant, it suddenly grows up, causes flowering. So basically gibberellin causes flowering, fruiting hormone. In short, you can write gibberellin is flowering and fruiting hormone. Gibberellin in short is flowering and fruiting hormone. Gibberellin breaks dormancy in plants. Gibberellin breaks dormancy in plants. So if you play in plants and seeds, let's say in plants and seeds are basically dormant, right? Plant, if it is dormant, then it has in some storage organ form, in bulb, rhizome, any other form, right? Tuber, all these things are dormant forms. Okay. So if you spray gibberellin, these effects will become reverse. Gibberellin promotes conversion of starch. Starch is insoluble food into soluble carbohydrates. Soluble or mobile carbohydrates. Mobile means they can move from one place to other. Starch is immobile. Starch cannot move directly. It cannot move. It can be converted into other things. Then it can move. So it causes mobilization of food reserves. Mobilization of food reserves. It is used in making the most common thing associated with gibberellin. The characteristic feature of gibberellin is bolting phenomenon. If you see an unknown substance which is causing bolting response in plants, it is definitely one and only thing, and that is gibberellin hormone. This question has been asked. An unknown substance when sprayed on a plant causes the plants to suddenly grow tall and produce the flowers on inflorescence axis. This phenomenon is called dash, and it is due to which plant growth regulator? Answer is bolting, gibberellin. Characteristic feature or response to gibberellin is bolting phenomenon. Bolting. Bolting in most plants. And what is bolting? I already told you what is bolting. This is bolting. Okay, this is bolting. Sudden development of flowers like this. This is bolting. Previously it was not having this response. Previously it was not having this response. Now suddenly it is undergoing flowering. This is called bolting. And other examples are development of seedless grapes, seedless all this that. So grapes, I have already given you a fair example. Like these are the large, you can see these are large juicy grapes, very very large varieties. So all these grapes are due to gibberellin hormone, spraying of gibberellin hormone. Okay. Gibberellin used to increase fruit size in grapes and other plants. Used to increase fruit size and sugar content. This is the last point. I know my time is over. Increase fruit size and sugar content in grapes. And other fruits as well, but grapes is most common. In the next turn, tomorrow we will complete this chapter. So class 11th portion will be completed from my side tomorrow, and in the next week we will continue with the last part of that, which is the remaining 12th part. That will complete in the next week. Okay students, so thank you for your suggestions. In the next class we will discuss a few more things. Let me know what else you need, the study resources and everything, okay, and have your lunch quickly because the next class is beginning. So thank you and have a great day. Thank you.